I see that it's about 20 minutes until the lunch break, and probably we are going to run into the lunch break, but no worries, it's 90 minutes. I mean, we don't spend 90 minutes usually for the lunch, I believe. How are you, by the way? Is it okay? I mean, so far so good. I see some tired faces. I see somebody almost falling asleep. Yes, it's you. Yeah, I'm looking at you. Yes. Now it will be more difficult to fall asleep. Okay, two more speakers before the lunch break. Aldis Butlers from Latvia State Forest Research Institute, Silava. Are you here? Yes. I was looking at picture of you in the internet, and then I was looking at you in the audience, and I thought, is that the same person? But it is. So the floor is yours.
Yep. Hello, everyone. So this presentation will be about main results of the OrgBalt project regarding carbon balance in forest lands, agricultural lands, including croplands, grasslands. And but I will start with introduction with key aspects of the methodology, because we have to understand how these emission factors were derived to better understand and have a more healthy discussion. So during the study of 2 years, we had 46 study sites in forest land, including drained and undrained sites. Undrained sites we had 7 in number, and in drained sites in total 19. With dominant 3 species of black alder, silver birch, Norway spruce, and Scots pine. And in grasslands and croplands we had only drained sites, in total 20. In grasslands there were only perennial grass. In croplands the crops were wheat, rapeseed, maize, and beans. And slightly more about the conditions in forest lands which were undrained. The mean groundwater level was about 10 centimeters, and in drained sites it was mean about 60 centimeters. Something similar in grasslands and croplands, where mean groundwater level was 50 centimeters and 78 centimeters, to better understand the conditions of the sites. About data collection, like Jurka already started to explain, we measured not only CO2 emissions, but we measured also carbon input into the soil. In forest lands it was foliar, fine litter, both above ground and below ground, grown vegetation of herbaceous plants, and fine roots of trees. And in agricultural lands, of course, it was above ground and below ground vegetation. Foliar litter fall we collected continuously during the 2 years with litter traps. You can see the one in the background of left top picture, the green one cone there. Grown vegetation we collected once per year at the end of the vegetation season. And fine roots of trees we collected using this in-growth core method, which is based on this, let's say, cylindrical form mesh bag with a mesh size 2 centimeters. We fill it with the soil from the study sites, put it into the ground up to 20 centimeters depth, and whatever of the roots grows into them on this core, we consider fine root production, and we assume that fine root production is equal to mortality. And regarding CO2 emissions, we had 2 approaches. We aimed to measure heterotrophic respiration directly by applying trenching method, where we, at the measurement point, we put the chamber on the soil, and their little layer and grown vegetation was removed. And also to exclude ingrowth of new roots, we use trenching. That means we cut the roots around the measurement point and put into the soil geotextile at the depth of at least 40 centimeters to limit the ingrowth of new roots. And also we measured total respiration. That means that we put the chamber onto the soil where the grown vegetation and litter layer is intact. That means in this case we measure not only heterotrophic respiration but also autotrophic respiration, which is CO2 emissions by living plants, including below ground and above ground plants, which were included in this headspace of the chamber. And also additional data like soil temperature, which was measured manually and continuously, and these continuous measurements were used to interpolate CO2 emissions by each hour during these 2 years by using relationships found between emissions and the temperature at the moment of the measurements. So this one is about approaches calculating soil carbon balance that can be called also emission factors. There were 2 approaches. You can see the yellow boxes. The top one is approach for calculating forest floor in the case of the forest land or agriculture ecosystem carbon balance. Another approach is aiming solely on soil carbon balance, on soil emission factor elaboration. For both of the approaches, carbon input approach was the same. In forest lands, what the biomass we measured, annualized biomass of the litter sources I mentioned, we recalculate it to the carbon content and apply it as a carbon input into the soil. In grasslands, the approach is similar. In this case, only above ground biomass and below ground biomass with additional assumption of turnover rate of below ground biomass. In croplands, it's a bit more complex. Without below ground biomass litter, we estimated also above ground and below ground litters by harvest residues, where we had to use another assumption about harvested wood products, So that's what we use for carbon input, and when we look at this, again, the yellow box above, this was how we calculate the carbon input. How about carbon output? We use directly measured this total respiration. That's how we derive this emission factor. Regarding soil carbon balance emission factor, we had to recalculate the total respiration measurements to soil heterotrophic respiration. But in our case, we used an empirical approach by using information from previous studies on the relationship between soil respiration and heterotrophic respiration to do this recalculation. We had to do this because our heterotrophic respiration measurements showed significant heterotrophic respiration overestimation. The main reason for that was because when we do the trenching, we cut the roots. The measurement time in the study is 2 years, and the decomposition time for the roots to decompose only half of the mass of the roots usually takes in our climate conditions about 2 years. And in our case, as our study sites were nutrient rich means sites are abundant with vegetation. That means we had high below ground biomass, and when this high biomass is decomposing, we have huge overestimation of heterotrophic respiration. By literature, heterotrophic respiration usually in our climate conditions is about 4, probably up to 6 in some cases, tons per carbon per hectare a year. But this overestimation alone induced around 6 tons of overestimation per year, considering we had the biomass in these sites of below ground up to 40 tons. Now let's get to the main results. We will have such 3 slides. On the right side, we see the mean values of total respiration, recalculated heterotrophic respiration values in the table, the mean input with the liter, and then when we sum it up, we see the forest floor, emission factor, soil emission factor, and later on I will get to drainage input factor. So let's see what happens in drained and undrained sites regarding soil emissions. The 2 bars in the graph on the left side, we see that in drained sites we had considerably higher soil emissions, but we also had higher carbon input by vegetation litter. So if emissions were higher for about 2 tons per hectare a year, litter in drained sites was higher for 1 ton per year, so we're not able to compensate for these carbon losses if we consider total respiration, but when we recalculate it to heterotrophic respiration, aiming to estimate soil carbon balance, we actually got results that we did not discover that the soils during the study period lost soil carbon stock. We see those brown bars on the left, drained and undrained, but uncertainty is high. Actually, they show removals, but safer to say that we did not actually see that the soil would lose carbon. And when we compare these 2 results in drained and undrained sites, assuming then when we combine those, we see the effect of drainage on soil carbon balance. We see that the negative impact was 0.43 tons per hectare per year. That means the drainage, in our case, drainage impact was reduced carbon sequestration. And when we compare it with the default IPCC emission factors, what we got in case of drainage impact, it's similar to the default factor for nutrient-poor soils in boreal zone, slightly lower than the nutrient-rich emission factor in boreal zone, but significantly lower than the temperate default emission factor. In case of the grasslands, here we can see that emissions were considerably higher than in forest lands, and the litter input was at least 2 times lower than in forest lands. Consequently, we have higher emissions, less carbon input. When we combine that, we see that the soil lost about 4 tons per hectare per year. And when we can get into more stratified data, because we had 2 classes of the study sites, one with deep organic soil organic layer and another one with shallow layer, something on the threshold of organic soil definition, then we see that actually these soils that had the shallower or organic layer had the higher emissions. And when we compare these results with the IPCC default emission factors, we see that these shallow soils had similar emission factors than IPCC boreal and IPCC temperate deep drained soils, and these soils in Latvia's case with the deep organic matter were more similar to temperate shallow drained emission factor, but more or less the emission factors are quite similar considering the uncertainty ranges. Regarding cropland, again, similar, if in grasslands emissions were higher compared to forest lands, and in cropland emissions are higher compared to grassland. Here we saw a total respiration reaching even 16 tons per hectare per year, and also in soils with a shallower organic soil layer. But in this case, the difference between deep and shallow soils were even higher than in grasslands. But we have to consider that uncertainty was also high, so actually mathematically, we couldn't say that it's a significant difference due to this uncertainty. And the carbon input in croplands, we estimated was quite similar than in grasslands. Consequently, the emission factor in croplands is quite higher than in grasslands, reaching up to 7 tons of carbon per hectare per year in these shallow soils, and in deep soils, something much less, 4 tons. Comparing to default emission factors, we see that emission factors are quite similar also to IPCC default guidelines, especially when we compare the shallow soils. But when we compare the deep soils, then emission factor is considerably lower. Yes. Yep. And it's time for discussion.
All right. You said it yourself. I mean, it's time for discussion. Why not? We have actually quite a few questions again. Thanks for that. Thank you. You see them? We can read it from. Yeah, but then you need a microphone. We can do it like that. You just read it out and you say the answer.
Yeah, probably. We can do like that.
Less work for me.
Yeah, actually, regarding grasslands, I believe we had both cases with plowing and without plowing, I think. And how it affects, actually, it was difficult to quantify because we had to make some assumptions regarding how the impact of plowing is doing, how it acts on the carbon input. But I will leave this question to Darta when we have the poster presentations up in the dinner time. lunchtime, and she can help you answer this question better. Yeah, the key difference between different emission factors, it's something I already mentioned. Mostly it's because emissions itself differs, and also carbon input differs. So in forest land, the carbon input by liter is considerably higher than in case of agricultural lands, and that's the reason why, and also the emissions are lower. When we put that together, that's the reason why emission factors in forest lands are much lower. And why these emissions are such high in agricultural lands, it's most likely due to this nutrient availability in the soils that promote microbial activity that increases these emissions. And when the carbon input is low, that's not able to compensate for these increased emissions due to mineralization. Yes, we had something revetted in OrgBalt sites, but actually they got flooded in this last season. Those sites were meant to be oversted lands with revetting, or let's say managed groundwater level, but we saw that the areas got flooded, and most likely the trees died there, so we will have to see what happens there. That's another reason why 2-year studies are difficult to quantify in 2-year studies, how the revetting actually acts in the specific areas, because we cannot predict how revetting will do in, let's say, 10 or 100 years, because our climate conditions or meteorological conditions are varying in each year. That's the reason previously we had mentioned in some questions, why do you gamble on afforestation? I actually would say we gamble more on revetting because it's more difficult to predict the groundwater level fluctuation. So we actually cannot say what will be there and what will be variation groundwater level in let's say 100 years. But in forest land, it's more easier to predict how the stand will develop. That means we can predict the carbon input by liter. We also can predict more or less how often happens these natural disturbances. So I would turn this comment opposite side, that revetting is probably even more risky if we take into account all the kind of risks that we can.
Okay, let's take the next one. The upper road has 3 likes.
Okay, upper one.
Yes.
Can you clarify if there are no case negative emission factors? Do you mean the default ones? Actually, there must be default ones, because when we look at the uncertainty range of default emission factors, we usually pay attention to the mean value. But when we look at the uncertainty range of default emission factors, we can see also negative values. That's the reason why uncertainty range can get also below the 0. So that's not correct. There are emission factors outside there also negative.
Okay, and maybe one last one, the upper one by Aldis, the most active, apart from anonymous, of course.
Yeah. The current approach comes from IPCC's guidelines, because emission factors we get there is actually emission factors from drained soils. But by idea, IPCC actually should report on anthropogenic impact of emissions, and that's not the same drainage impact emission factors or drained soil emissions. Because when we should report anthropogenic impact, we should take into account the baseline, which is undrained soil, and compare it to the drained soil, and that's anthropogenic impact. So my suggestion would be that we should move forward in this methodological improvement to actually report what we should report: anthropogenic impact, meaning we should report drainage impact, not emissions from drained soil.
Are you here till the end of the day, by the way?
Yes.
So I suggest you approach Aldis for your questions that were not covered through the lunch break. Thank you so much. All right.